Yeah. Good morning. So today we are going to learn enzymes. What is the definition of enzyme? Uh, what are the classification of enzymes? Then we will discuss factors affecting on enzymes. So three parameters we will learn. That is, first one is definition of enzyme. Second one is classification of enzyme. The third one is factors affecting on enzymes. So first of all, we want to know what is the definition of enzyme. An enzyme is a biocatalyst which can fastens the rate of reaction. That means which will increases the rate of reaction. In presence of enzyme only, the rate of reactions will be proceeds. So that is the one condition. Actually, what is an enzyme and where we found enzymes? Enzymes are those they can synthesize in the body. So the properties of an enzyme. Enzymes are protein in nature. That means we have the one definition. All enzymes are proteins, but all proteins are not enzymes. So in the same concept, here when we learn enzyme, the nature of enzyme is a protein and it is a colloidal in nature. That means they collide with each other to form their specific reaction, and they are thermolayable. They will resist over the temperatures up to the extent. That means a maximum temperature, there is a denaturation of enzyme activity, or if there is a low temperature also, the specific function of enzymes cannot see. Even enzymes show the specificity on their action. That means enzyme, you take carbohydrate, that means starch, amylases will work on carbohydrates, lipases are there, and these lipases work on fats. So that we call them the specificity of an enzyme action. Sometimes even in the nucleotide, RNA, DNA and RNA. Apart from these, RNA is also referred as acting like a catalytic in nature. So such type of RNAs are known as ribozyme. So this ribozyme is nothing but an RNA which showing the catalytic property. So such type of RNAs are known as ribozyme. When you see here, there is an equation how enzyme kinetics will work. We will discuss in enzyme kinetic class, but here we will have one idea. So enzyme equation, we have the two different steps, step 1 and step 2. In this step 1, enzymes react with a substrate to form enzyme substrate complex, and which one is a binding step, which is a slow step. Whereas after the formation of enzyme substrate complex, it will react with enzyme and substrate to proceed the product. So which is in a second step, and which we call them as a catalysis step, which is a second step, and it is the fastest step. So enzyme kinetics, in this enzyme kinetics we will learn about the different types of steps and their action, and enzyme kinetics and Michaelis-Menten equations and their property we will discuss in upcoming classes. So as we learn, an enzyme is a biocatalyst. It shows the property that is a protein in nature, colloidal in nature, thermolayable in character. It shows a specificity in their action. And even DNA, RNA, some type of an RNA also acts like an enzyme activity because they have the property by the name catalysis. Then now we will go with the classification of enzyme. Here we have the 6 classification according to IUP classification. So here we have oxidoreductase is the first enzyme classification. The next one is the transferases. The next one is hydrolases, lyases, isomerases and ligases. So these are all the 6 classes of an enzyme which they are going to be show their enzyme activity. So first one is oxidoreductase. Oxidation reactions, right? So oxidation and reduction, maybe addition of hydrogen group or reduction of an hydrogen, removal of hydrogen group. So here we have the 2 enzymes, namely oxidases, which involve in oxidation, hydrolases, which involve in removal of hydrogen molecule. So we call them as these group of enzymes which helpful in oxidation, and these group of enzymes which are also involved in reduction process. So such type of an enzyme were placed under the class of oxidoreductase. Then another enzyme we have transferases. The name it indicates transfer. Transfer of what? The transfer of molecules or transfer of functional groups. There may be remove amine groups, there may be remove carbon dioxide in their reaction. So such type of enzymes are placed in the class by the name transferases. So enzymes are transferases and kinases. These two enzymes are placed in a group, we call them as a transferases. The next one is hydrolases. Hydrolases, the name it indicates hydrolase, in presence of water, they show catalytic, that means hydrolytic cleavage. They remove the water molecule in a reaction, enzymes combine with the substrate, they remove hydrolytic groups. So in this reaction, which enzyme is responsible means hydrolases. It will work removal of water molecule cleavage. So here an enzyme by the name nucleases as well as proteases. These two are placed under the class of hydrolases. So they will ask in examination point of view, nucleases and proteases are grouped in which type of enzyme classification? Oxidases, transferases, hydrolases, liases or ligases they will ask. So in terms, you will get concentrate that nucleases and proteases are group of hydrolase enzymes. Are you getting me right now? Another one is liases. Liase means addition of atom or removal of atom. For example, CH3 double bond, CH3 single bond, CH3. Removal of water, removal of hydrogen group here. So in terms, what will happen? C double bond, that means converting into to form the double bond itself. So that we call them as liases. Addition or removal of an atom, so results in the formation of single bond to the double bond, maybe a triple bond. So that we call them as liases. So here what type of an enzymes are used means, here we have decarboxylase enzyme and aldolase. Decarboxylase and aldolase. Then another enzymes are there, here is isomerases. Simple. In terms, in chemistry also we go through that. The isomerases are those enzymes which convert the cis configuration into trans. Cis configuration into trans, or a trans is going to be converted into trans are going to be converted into cis, or cis are going to be converted into trans. So such type of geometric changes or structural changes can be done through isomerase enzyme. Example is isomerase. And here in terms of the last enzyme in enzyme classification, that is ligases. This is a ligases are those enzymes which frequently used in molecular studies or in DNA fingerprint studies, for even in PCR technology. That means joining and joining of nucleotides, maybe a DNA. In terms, when the two DNA molecules were cleaved, for example, this is a DNA molecule, here it will cleave. So the cleaved DNA can be joined in presence of an enzyme DNA ligases by phosphate group, addition of phosphate group and OH group. Alkaline phosphate is an enzyme which is responsible to join phosphorate group, and to this compound there is an enzyme by the name DNA ligases. They will help the two molecules to be joined. In presence of this joining, ATP molecules are required. So another one. So this is a classification of enzyme. The first one is oxidoreductase. It involves in oxidation as well as a reduction reaction, addition of hydrogen or removal of hydrogen. Transferases are those, the transfer of molecule or transfer of functional group. Maybe any functional group may be changed. So here hydrolases, in presence of hydrolytic enzyme or hydrolytic cleavage, the molecules can be changed. Liases are those, they helpful in addition and removal of atoms from a molecule to convert it into double bond or triple bond, etc. Then isomerases. Isomerases are those enzymes which are convert cis into trans conformation, trans into cis conformation. Ligases. Ligases are those, they helpful in most of molecular studies. The ligases, especially the DNA ligases, are helpful in PCR technology, DNA fingerprint, or molecular studies, any diagnostics or any DNA fingerprint technology. So in case the two molecules of DNA can be joined in presence of DNA ligase. In this reaction, ATP is mandatorily used. Then factors affecting. The factors play an important role in enzyme activities. So the first one is enzyme concentration of an enzyme. In this concentration, if more enzyme will be there, the reaction increases the reaction rate, and this increasing the reaction rate should be proportionally increased. If there is a maximum substance, minimum enzyme activity, the reaction will be slow. Maximum substrate, maximum concentration of enzyme, it will proceed increasing the rate of reaction, which is proportionally increases. The second one, concentration of substance. If the substance is more, increasing the velocity of enzyme is proceeds more, but until unless it reaches the maximum extent, it will decline. So that is the concentration of substrate. If more substrate will be there, if less enzymatic activity, in due course of time what happen, enzyme activity will be decreases. So concentration of if more product will be there is a decreasing in enzyme velocity. More product, less enzyme activity results in the formation of decreasing in enzyme activity. Temperature. All enzymes cannot be active at a particular temperature. It should have optimum temperature. 40, 42, 45 degrees centigrade is an optimum temperature for our different enzymes. So in this area, increases enzyme velocity up to optimum temperature. If more temperature will be there, again there is a declination of the reaction. In the same way, pH. pH is also have increasing the activity during pH 6.8 in some cases, or in a higher organism maximum enzyme will be worked on 6.8. But if pH will be more and more, again there is a declination of activity will be increases. That means there is a slowdown of enzyme activity will be there. Even enzymes and cofactors, magnesium chloride, manganese, zinc, copper, calcium, all these cofactors are which are activator, which are facilitating to activate to perform an enzyme reaction, also play the factors for affecting enzyme reaction. Even time, ultraviolet lights, radiations, oxidative agents, some sort of an anti-enzyme, looking similar but performing against to this enzyme activity, such type of an anti-enzyme. These are all the factors which will affect the enzyme activity. So this is today's class regarding with enzyme definition and how the equation will be worked, enzyme classification, what type of an enzymes and their example should be mandatory, should remember. In terms of an examination point of view, they will ask questions here, and even in terms higher order thinking questions, they will be framed how the factors affecting enzyme activity during the reaction, right? So this is today's class.